So now let's dive a little deeper inside of the cell. So figure 3-1, right at the beginning of this chapter, again does an amazing job of kind of summarizing pages and paragraphs full of information, right? This should be, again, sorry I sound like a broken record, review from Bio 1111, but a chance to remind yourself what all of these different organelles do. Now, remember, an organelle, this is actually kind of cute, right? It's like a play on words, right? An organ, we know, is a group of tissues working together to achieve some function. And now this is like a little, like, organelle, a subset of the cell that's achieving a certain function to benefit this cell as a whole. I would like you to know the function of these organelles and a couple other structures they're showing, like microvilli and cilia, at the level of this image, right? So you don't need to go any further than that. And really what I want to do is I'm going to go ahead and focus in on a few more like processes and show you what organelles are involved there, and spend a little more time on those particular structures, but do make sure you look this over and understand how these structures work. So what I want to jump into is this process of protein synthesis, how a cell takes your genetic code, right, the DNA, the instructions, and actually ends up with this end product, a protein, in this case a protein that's being packaged for export. So we'll see protein synthesis, packaging, processing and packaging. So we have a variety of different organelles involved here. We're going to start in the nucleus. We'll talk about the function of ribosomes, the rough ER Golgi apparatus, and so we're actually covering a few of the big ones here. So let's take a peek here. All right, so we're inside of the nucleus. Inside of the nucleus is where the DNA is packaged. We'll talk a little bit more about that structure, right? But remember, your DNA is this double helix, and the key thing is that it codes the order of letters in the DNA codes, it's the instructions for how to build a protein. Now, most of your DNA strand is actually not particularly useful. We call it non-coding DNA. It's something like 2% that actually has genes. It actually has certain instructions for certain proteins. Okay, so we're going to begin with this step that you might be more familiar with the term transcription. In transcription, we're going to take that DNA code, we're actually going to unzip the DNA, right, pull it apart, and make a copy of one side. So it's like a template, right? The cellular machinery is going to read one side and put in all the complementary bases, but we're going to build a structure that, instead of being a double helix, is a single strand, slightly different molecule that we talked about in chapter 2, and that is RNA. This is a specific form of RNA called messenger RNA that is going to go into our next step, right? But transcription, you need another N in there. There we go. Transcription, DNA to RNA. Okay? That RNA is actually going to leave the nucleus. There's actually little proteins, right? Again, it's a selectively permeable membrane. This is going to allow RNA out of the nucleus, but not into the nucleus. So some people were concerned about the messenger RNA vaccines for COVID somehow actually changing our DNA. But these are one-way proteins. So that messenger RNA leaves the nucleus, comes out here into the cytoplasm, and finds a ribosome. Ribosomes are where translation takes place. And translation is the process of taking that RNA, reading it letter by letter, and building a polypeptide chain, a string of amino acids, which will become our protein. All right, so RNA to protein is translation. Now, if you have trouble because these two words sound the same, think about this. DNA and RNA are both nucleic acids. They're the same language. So in transcription, this would be like a courtroom transcriptionist sits there and types down everything that's said. Whatever language, right? If it's English, she's writing in English. Or he is writing in English. Soon that's probably AI. Maybe it already is. Okay, but it stays in the same language, the language of nucleic acids. Translation, we're switching languages. We're going from the language of nucleic acids to amino acids, right? And so kind of clever descriptions here of this process. Okay, now this growing polypeptide chain, what they're pointing out here, sorry, sometimes those ribosomes are free-floating in the cytoplasm, and that's pretty typical. If the protein is going to stay inside the cell, typically the translation will happen on a free ribosome. If it's meant for packaging and export, typically that is going to take place on what we call the rough ER, right? The rough endoplasmic reticulum. This is endoplasmic reticulum that is studded with these ribosomes. And so here we see messenger RNA that has come to that ribosome, and we see the polypeptide chain actually being built inside of the endoplasmic reticulum. Okay. Again, we see this polypeptide chain or protein being developed inside the endoplasmic reticulum, and it's going to move through the endoplasmic reticulum and actually be packaged into these vesicles that then move over to the Golgi apparatus, right? So we see a little bit of endocytosis there into the Golgi apparatus. The Golgi apparatus we're able to actually modify, so if it was, say, a glycoprotein, if we need to add a sugar on or something like that, we change that protein, we repackage it, and send it through a lysosome to the cell surface where it can actually be exported, right? So this would be, I'm trying to think of an example, maybe the way some of your hormones are produced, or maybe some mucus is produced for export. Okay, so transcription and translation is the process of actually building the protein, and then we're going to use the endoplasmic reticulum and the Golgi apparatus. Here we go. This picture actually makes way more sense. The endoplasmic reticulum and the Golgi apparatus to package it for transport. So again, DNA, RNA, heading out to ribosomes. Ribosomes on the rough ER are able to package that protein. At the Golgi apparatus, we can modify it, repackage it, and export it from the cell. This is kind of a cool example here, right? So the Golgi apparatus, some of what it's making are called lysosomes. So that would be on that original picture of the cell with all the different organelles. And these lysosomes are really meant to break things down. Sometimes it's something that the cell has brought in. It'll fuse it with a lysosome and get it to digest it. In this case, they're actually showing a damaged organelle basically being kind of ground up and like those waste products being released. Here we see a substance actually being brought into the cell, that fusing with a lysosome and being able to digest it. Maybe some of those substances we want, maybe some are actually going to get dumped as well. Okay, a couple other key organelles. The mitochondria, you all know that the mitochondria is where ATP production takes place, right? And ATP is that cellular currency of energy. When you eat carbohydrates or proteins or lipids, they have calories, right? That calorie is just a measure of energy. The energy from that lipid gets transferred into bonds in the ATP. We don't turn lipids into ATP, we just transfer the energy into a form that cells know how to use. Chapter 25 in your textbook So a topic of A&P 2 will go through this process of cellular respiration, and hopefully you got a good sense for that in Bio 1111. Okay. The last organelle that I do want to touch on is the nucleus. And I want to talk about the nucleus, right, in this idea of genetics, right, where this DNA or genetic code is housed. And so hopefully we're getting the structure of DNA down a little bit from the macromolecule discussion in chapter 2. But just a reminder that when you look at base, what we call base pairing, G's always go with C's, A's always go with T's. And I like this image, they're showing dotted lines down the center of the DNA. This is showing a weak bond between the two strands of DNA. Would that be a covalent bond, an ionic bond, or a hydrogen bond? If you said hydrogen bond, you're correct, right? So we're able to peel these apart, stick them back together. And that's why you can open it up and do transcription, right? Make a copy, make that messenger RNA, and then it goes ahead and heals itself. So they're kind of showing you that again in a bit more detail. Another thing that they are pointing out is when that messenger RNA goes to the ribosome, recall from hopefully Bio 1111 that message is read 3 letters at a time. So we call those 3 letters a codon. And here they're showing when that messenger RNA makes it to the ribosome, the way this works, right? We have a codon that actually indicates to the ribosome, hey, this is where the protein starts. Start here, right? And a codon on the messenger RNA has a complement, right? Notice now we're in RNA, so A's go with U's, A goes with U, C still goes with G. This is transfer RNA. It's another form of RNA, but in this case it's transferring amino acids to the ribosome. So as we read this code 3 letters at a time, it tells us which of the 20 amino acids get brought to the ribosome and put into that polypeptide chain. Now, the ribosome is technically an enzyme. Some people actually call it a ribozyme to indicate that it's an enzyme. It's going to bring these 2 adjacent amino acids close enough together to encourage the formation of a peptide bond between those amino acids, right? Close enough proximity, we catalyze that reaction, okay? And we just keep pulling this messenger RNA through like a tape, and different transfer RNA molecules keep coming. Now, the cool thing about this, right, is then we're making this polypeptide chain. And your genes, right, the 25,000 or so genes that you have code for all the different proteins in your body, right? How to make the keratin in your hair, how to make the myosin in your muscles, how to make the enzyme lactase, right? All of these things are in your DNA as instructions that then the cellular machinery is able to build. As long as you have the correct set of amino acids and the correct instructions, this goes pretty well. When that doesn't happen, we can see a variety of different disease states, right? So here we are at the most minuscule little part of the code where we even said people who are lactose intolerant, it's because they don't have the genetic code to keep making this enzyme lactase, right? Or if you think about sickle cell anemia, there's a mistake in the DNA that's going to make it so hemoglobin in red blood cells doesn't form properly. You think about cystic fibrosis, there is a single amino acid that is incorrect in a chloride transporter on a cell membrane in the lungs, right? And that causes this mucus buildup and a disease called cystic fibrosis. So your genetic code and our cells' ability to build those proteins in the code becomes extremely important. Okay. In lab, I know you were looking at the cell cycle again, should be review. The thing that we want to make sure that you really get down and understand and keep a hold of this time is that in the cell cycle, right, most of the cell's life cycle is spent in interphase. And interphase is where real life happens, right? If this is a liver cell, it is spending interphase doing liver things. If it is a muscle cell, it is doing muscle things, right? Mitosis, which gets all the attention, is really just when the cell is dividing, right? And what we'll see as we move through different organ systems is that some cells are much better at undergoing mitosis than others. And so this will affect kind of rates of repair, rejuvenation, things that we can do in different tissues, different organs, and organ systems. So I won't belabor this, but do make sure you go back through and review your mitotic stages. For lab, let me know if you have questions on those. Okay. This one's just showing cancer. So since I pointed it out earlier, right, just a couple things here. When you look at cancer, you are dealing with a cell that undergoes very rapid mitosis, right? And kind of escapes these normal rules of how cells behave. So typically what happens, so here's kind of your application of mitosis, right? If you scratched yourself, you would want that injured area to refill with more skin cells, right? And so you want that cell to undergo mitosis. What typically happens is you only get as many cells as are needed for that space, right? Then the cells are sending messages to each other that say, cool, we got it, there's enough, right? We're holding hands with our neighbors, we're good. Cancer cells ignore those messages and they will just pile up on top of each other, right? That's how we get something like tumor formation. Most of our cells follow rules about how many times they can undergo mitosis, and cancer cells ignore that. In fact, they have a line of breast cancer cells, I think since the 1950s, that just keeps dividing and dividing and dividing. Very abnormal. And then the last thing is one that we talked about earlier, this idea that those cells don't even have to keep holding on with those cell adhesion molecules, keep holding hands, and instead they can metastasize, they can move through the body, land in distant locations, and create tumors there as well. So that was just this idea of telling you a little bit about cancer, a little teaser, is to think about why mitosis is important and how maybe mitosis gone wrong can actually be really bad. We'll see you in class.